Hi again, everyone. Welcome to Chapter 6, Part 3. Here we're going to discuss in this section of the chapter a little more detail about chloroplasts, peroxisomes, and cytoskeletal elements. Very, very important and often overlooked in cellular function. We're going to be picking up where we left off from last time, this time focusing on chloroplasts first and then peroxisomes in a little more detail. We've already mentioned it, but we're gonna break these down a little bit more. The main point to remember about peroxisomes is they're going to help break down reactive byproducts from either the mitochondria and or the chloroplasts. Most all of you probably know, I'm sure, that chloroplasts contain a green pigment, and so we have the beautiful green leaves in the summertime. That pigment is known as chlorophyll, but there are any number of other types of enzymes and molecules that are involved in photosynthesis. So we find the green chloroplasts, but we also find other colors found in what are called plastids. They are like chloroplasts, but they contain slightly different pigment colors. And that's why we see these colors appear in the fall, because chloroplast is being broken down and recycled for other biomolecules. And what we see left over are the plastids, so things like xanthophyll and carotene and anthocyanin. Those are all like chlorophyll, but they are absorbing other wavelengths and help protect the cell that's absorbing the light, the cells in these leaves, from additional damage due to the light coming from the sun. And the arrangement of a chloroplast and the other chloroplasts-like plastids is this double membrane situation, a lot like in mitochondria. But unlike the inner fold that we have in the cristae of the mitochondria, we have these sort of stack-like structures that are somewhat interconnected that are called grana, grana being plural. And we have a similar sort of inner matrix, very, very rich fluid and so forth. This membranous structure, by the way, closely resembles the membranous structure that we have in the photoreceptors of our eyes. So very, very interesting morphology similarity there. These thylakoids are these membranous sacs, and when they stack up, that's what we mean by a granum. And then in plural would be grana. And then we have this internal fluid, and again we have this space between the inner and outer membrane. And so here's a drawing, and then a photograph or an electron microscopy image, and then a high resolution microscope image. We're going to talk a lot more about chloroplasts when we get to the photosynthesis chapter. Moving on to peroxisomes. Peroxisomes are specialized structures. They're kind of weird, and here's why. They have this phospholipid bilayer, and then they have this interior, very, very rich protein. It's not a matrix per se, but it is a very, very strong solution of proteins. Sometimes so strong that you actually get proteins crystallizing out. Proteins can and do crystallize, and that's actually one of the ways that we study proteins, is by crystallizing them and then blasting them with electrons. But in any case, in the older ones, you will often see this crystalloid core. Is it a vacuole? It's hard to say. We just don't really know a lot about these. We're just now starting to get a handle on them a little bit. We know that peroxisomes contain enzymes that will break down hydrogen peroxide and other what we call reactive oxygen species. Reactive oxygen species are molecules that include oxygen. There's even reactive nitrogen species as well, and they will render them less reactive. What we mean by reactive is that they will form chemical bonds with lots of different things, including biomolecules. And when you bond biomolecules, you change their shape. And when you change their shape, you change their function. And creating ATP and doing the electron transport and doing photosynthesis, you often get these ancillary reactive oxygen species occurring because you're shuttling electrons around. And then they can potentially threaten the cell. And so your cell has very robust antioxidant capacity. And you might hear about antioxidants in diet, and I do have some things to say about that, but I'll save that for later. In any case, these things also do other reactions with different functions. We're going to talk about that in a subsequent slide. It's not just reactive oxygen protection. How they're related to organelles, like I said, are they vacuoles? It's some sort of specialized vacuole? We're still working on that. So peroxisomes are involved in taking alcohol and making it less toxic. Other forms of detoxification, reactive oxygen species. You see ROS right here, ROS metabolism. ROS stands for reactive oxygen species. Here's an enzyme catalase, and there are other oxidases in here. We will talk about beta oxidation later on when we talk about ATP synthesis. Beta oxidation is basically the process of breaking down fatty acids and turning them into acetyl-CoA. It's the way that we can use fat to create ATP. So beta oxidation, and then another one closely related is alpha. That's just telling you which carbon bonds they're breaking down, by the way. So the alpha is at the beginning, and then the beta is the next one over. In any case, we're going to detoxify compounds. About half the alcohol that you would consume is detoxified. Your body does synthesize alcohol accidentally, and so your cells are able to detoxify that. So big time fatty acid chain oxidation, that beta oxidation process. And here's a picture of a peroxisome, and it's actually closely associated with this chloroplast, this mitochondria in here. That's because both of these organelles will produce reactive oxygen species that need to be detoxified. Moving on now, we're going to talk about elements of what we call the cytoskeleton. Like a skeleton, it can be relatively rigid and confer shape and structure and strength in some cases, but it also can serve other roles, including transportation and what I call compartmentation, that is to say, keeping things where they need to be in 3 dimensions. And so it's a network of fibers. It can grow and shrink dynamically. It's a very dynamic thing, more so than, say, bones or other connective tissue in our tissues. But it does organize in 3 dimensions. It can anchor, provide a basis of movement and that sort of thing. 3 major structures that we would want to deal with. The first being the microtubules. This is the largest one. It's also the most complicated in terms of its structure. It's kind of a helical molecule made of a protein called tubulin. And tubulin has a polarity to it. It actually has a positive end and a negative end, and certain motor proteins can move specifically towards a positive end, and some can move specifically towards a negative end. So microtubules made of tubulin. Then we have the smallest one. This is what we call microfilaments. It is made up of actin. It's a G-actin, if you will, and then when it forms these polymers, that makes a filamentous or F-actin. And then thirdly, these are the ones that you are probably in contact with the most. That's because your skin is loaded with it. Your hair, your nails are all made up of intermediate filaments made up of keratin. Keratin is the most common protein in intermediate filaments, and the density therein gives us the nature of whether it's rigid like a fingernail, or maybe a little more supple like a hair, or it can be networked like the keratin in your skin. And here's a lovely picture showing different kinds of cytoskeletal elements. These green things are actually tubulin. The red ones are actin. We have this kind of cortical actin. The cortex is the outer layer, and usually on the inside of the plasma membrane you have kind of a layer of like a mesh of actin. But these tubules serve as transportation highways for transport of molecules, proteins, and vesicles. Like I said, it helps support the cell and maintain shape. That's probably the one that I would least ask you about. I mean, that's pretty obvious. But we want to think about what would be incredibly important for a cell, which includes motion, motor proteins. The beating of the flagella in a sperm is where you have dynein motor proteins interacting with a microtubules to create a spinning motion for that flagella. And those vesicles can travel along these things. Very, very important for neurologic function because you have very, very long axons, which are subcellular structures supported by intermediate filaments, but also provide transportation so that you can send neurotransmitters and things way, way distant, sometimes more than a meter, which is thousands of miles in cellular terms, out to those distant ends of those axons. And that the cytoskeleton may also be involved in signaling. And like I said, that three-dimensional regulation, it means that you can bring things together that will react, or you can keep things apart that will react. There's a lot of new evidence coming along as our techniques have become better and better at isolating cytoskeleton and what's hooked up to it. I can't show you this video in this time here. At least I don't think so. I'll try, but I don't think you can. But I will show it to you in class, and it was filmed at 1 frame per second. And these are literally microtubules growing and changing in real time. Then we hit it with an inhibitor, and then it miraculously regenerated. In fact, that was something we never expected. And so we actually published a paper about this regeneration. But in any case, if you can't see it, then we'll show it to you in class. I'll double check. So here are those microtubules. Again, it's not showing you the helical polymer, if you will. You can almost make it out here in the figure. But motor proteins are specific for one direction on the microtubule. I have a GIF that I'll show you in the subsequent slide that will show you how this movement works. But these motor proteins literally walk on the surface of these microtubules using ATP to help change the configuration and produce movement. And as promised, here's our microtubule, here's our motor protein, and it's hydrolyzing ATP in order to achieve this change in protein conformation. So the ATP actually loads a spring inside of these motor proteins, and then the spring pulls back, creating motion. But here it's hooked up to a big vesicle with some proteins embedded in it. It's moving molecular cargo, and you can see another one out here in the distance. So the things I want you to know about cytoskeletal elements are which one's the biggest, which one's the middle, and which one's the smallest. So the microtubules are the biggest, the intermediate filaments are the middle, intermediate, and the microfilaments are the smallest. And then what their primary protein is: tubulin, actin, and keratin. These can vary, but keratin is our poster child for this one. And then a basic function or two. Again, structure is not going to be part of a correct answer. For instance, it involves chromosomal movement and cell division. When we talk about the mitosis and the cytokinesis, that's driven by the motor proteins and the microtubules. Actin filaments, muscle contraction's a good one. Cell motility, like in a pseudopodia, that's where a cell will stick out a projection of its cell membrane, like a foot and so forth. But also in part of mitosis and cleavage furrow. So when the cell begins to stretch and split, the actin will start to squeeze the middle, making it look like an hourglass at first, and then finally pinching it off. Intermediate filaments, the main thing I would tell you here is hair and nails and skin, or it's also the main protein that you find in the nuclear envelope, that nuclear lamina, if you will. And here's just a zoom in of tubulin, restating what I've already told you, so I won't belabor that. So again, some functions, the main proteins, and it's the largest of the three. Microtubules originate out of this structure known as a centrosome. A centrosome is composed of two centrioles, which are microtubule structures along with proteins. And I guess you could consider this an inclusion. I hadn't mentioned it before, but it's not membranous and it's a major functional entity in a cell. So I guess we would call that an inclusion. And these two centrioles are actually at sort of 90-degree angles to each other. And they're made up of this sort of 9 triplet kind of a star shape, if you will. So each of these is a microtubule, and there are 3 of them, and they create this sort of 9 structure arrangement that you can sort of see here. It's hard to kind of see it, but that's what it is. So a centrosome is composed of 2 centrioles, and these things can and do grow and project microtubules out from them during a couple of different times, during normal cell processes and especially during cell division. So here's a centriole. You can kind of see the triplet, 9 triplet thing going on there. Microtubules are very, very important for movement. Cilia in, like, for instance, your airways. Have you ever been near somebody who's a chronic smoker and you kind of hear them go, they're always kind of clear in their throat all the time? That's because the smoking immobilizes at first, but then completely destroys the cilia, which are present inside your airways, which are meant for clearing out debris and accumulated dust and things. That's why every once in a while you kind of have to, is because what's happened is material is built up there right by your epiglottis, and you clear your throat and then you swallow. Good for your immune system, by the way. But flagella is the same thing. They're microtubules. Microtubules driven by motor proteins that are fixed, and it creates a spinning motion. And here's that 9 triplet configuration. I guess I'm hesitating a little bit because there's also a doublet type configuration. They serve sort of different motion. You know, this one's more immobilization, and this one's more for that creating the motion, if you will. And what happens is you get this sort of, I guess the best way to describe this is if you take your hands and put them together like you're in prayer, you know, like in the prayer pose, and pull on one, and you'll see that your hand will pull in that direction where you're pulling down, you know, like you're making a fish movement. That's kind of what's going on here, is you get this pull in one way and it pulls the flagella or the cilia in one way, and then you pull the other way, so it's that kind of wiggling motion that you do with your hands. And we're probably beating this to death a little bit, but that's kind of that, like I said, if you put your hands together like this and you pull on one side, it's going to cause a deflection. You pull on the other side, it'll cause a deflection. You sort of alternate through there. Again, using ATP to change the shape of the protein, which then imparts force on whatever it's hooked to. And we'll finish off this segment with a couple of moving images for you. There's that flagellar motion of dynein with microtubules, and we have some cilia here. And there's just a motor protein showing you how they use molecular modeling techniques to figure out how these motors do their thing. And the reason there's a huge amount of interest in this for any number of reasons. First of all, male infertility, for one thing, but also in industry, they're really, really interested in this idea of being able to generate using little machines to do super small, super fine type of molecular level work. So there's a huge amount of potential money in being able to harness these things to do, you know, like little robots, you know, nanotechnology, if you will. So thanks for your time, and I will pick up again on part 4. Thanks again. Bye.